Good afternoon, guys. Mr. Luna here, just stopping by to share another video lesson with you. So today we will be covering the biological basis of behavior. More specifically, we'll be covering neurons, and we'll try to get another slideshow going on. So I'm going to try to squeeze as much as I can in the next 12 to 15 minutes, so that way we can have an efficient review session. I also want you guys to take notes as we're going through this. Don't just sit back and listen without taking note, because it's very important that you take note of the things that you do not know, and especially those things that trouble you, so that way you can question me, or that way you can start engaging in trying to learn those concepts. So without further ado, let's just jump right onto it. So the nervous system, right? We've covered that the nervous system is specifically divided into different divisions. So let's start off with the very foundational. So I need to tell you that the brain communicates through a series of electrochemical signals, which are passed through the spine to the rest of the body. Most communication are sent to the brain through the central nervous system, spinal cord, and fluids, which are then processed and interpreted by the brain. The majority of the sensory information sent to the central nervous system remains on the edges of the body's extremities, the skin, the bones, the muscles, the tissues, etc. So this is known as the peripheral nervous system. The sensory nerves that connect the central nervous system and relay messages to the brain. The brain regions, lobes, and cortical areas all interact to make decisions and send signals. So when we talk about the central nervous system, we're talking about the brain and the spinal cord. When we're talking about the peripheral nervous system, we're talking about everything else that is not the brain nor the spinal cord, but that relates to the nervous system and allows us to interpret feelings and things of that nature. So moving on to the other slide. Now, the somatic nervous system enables voluntary muscle movement. So I would write this down. Somatic nervous system, right? It enables voluntary muscle movement through motor neurons once commands are sent from the brain. Motor neurons are not able to be confused with sensory neurons that relay signals to the brain from the sensory receptors sent to the brain through the central nervous system. So the autonomic nervous system, this is another division. If you notice, this was the somatic. We just finished speaking about it. Now we're speaking about the autonomic nervous system. The autonomic or automatic nervous system is, however, largely uncontrollable and functions on autopilot, beating the heartbeat or digesting food or moving organs. Two functions of the automatic nervous systems are the sympathetic nervous system and the parasympathetic nervous systems. So the automatic nervous system is divided into other two categories: the sympathetic nervous system, which arouses, and then the parasympathetic nervous system, which soothes back down. So remember, we've just covered the central nervous system, the brain, the spinal cord. We covered the brain, the peripheral nervous system, which is everything aside from the brain and the spinal cord. We've covered the somatic nervous system, the automatic nervous system. Now we're moving on. So neurons. Within our nervous system, obviously we have neurons, right? The brain and all nervous systems operate with neurons, and they receive and send electrochemical signals to communicate. Neurons are made up of about 4 to 5 main parts. The soma is right here. The axons. So the axon is the little canal. Then we have the dendrites. The dendrites are over here, and these are the terminal ones, and these are the dendrites. And so we also have the synapses, which are the tiny junctions between neurons. So there's a little gap between all neurons, and that is what they call the synapse. So as I mentioned, the soma is a cell body which contains all the DNA and all other essential parts for keeping the neuron alive. Electrochemical messages are sent through the axons, which are covered by myelin sheath. Myelin sheath is just like a fatty substance that covers and protects the axon, and also allows the impulse or the transmission of messages or impulses to be a lot faster. So let's move on to the next one. So this is just a picture. It's a zoomed in picture of a neuron, but I think at this point everybody should know what a neuron looks like. We have the soma, or the nucleus within the soma. We have the dendrites. Then we have the myelin sheath covering the axon and the axon terminal. Remember, axon terminal are also known as the terminal buttons. This is where the information is sent to another neuron. The neuron receives it via the dendrite, and then the message is transcribed in the nucleus within the soma and is sent out to the rest of the nervous system. So let's move on. Now, neuron communication, right? When electrical charges travel through the axon, they reach the end and trigger the release of certain chemicals called neurotransmitters. So once the message does get triggered and is sent down the axon, once it comes out through the terminal is what we're going to start talking about. So this becomes an electrical chemical impulse where outside the axon are sodium ions and inside are the potassium ions and proteins, creating negative charges inside and positive outside the membrane. To send signals, electrical charges open the membrane, and this allows the ions to balance out, activating each other down the line. So once the signal has been sent, proteins in your body activate sodium-potassium pumps to respirate the sodium and the potassium. This process is known as action potential. When an electrical impulse passes through the axons of the communicating neuron, while all signals are the same strength, your neurons communicate in frequency of these signals like Morse code. So it's pretty cool the way we communicate. It all begins at a very, very tiny, minute level, and it ends up becoming speech and communication and whatnot. So this is what we're talking about right now. This is a sodium-potassium pump. You can imagine this right here, this blue area, as the terminal button, right? It's opening up, and this is where we have those terminals opening it up or closing, depending, right? And then we have the sodium-potassium pump binding the 3 sodium ions on the molecule of ATP. And then this is also splitting the ATP that provides energy to and change the shapes of the channel. So in other words, remember, we mentioned that sodium ions are inside the potassium pump, and then we have the sodium are outside and the potassium are inside. So potassium is inside and sodium is outside. And when we do trigger this action potential, they balance each other out. So they flow in and they flow out. So here you can see the potassium flowing out, and you can see the potassium flowing in and the sodium flowing out. Now, when we talk about reuptake, we're simply talking about the frequency of the signal in your body and how it releases neurotransmitters. So some of the neurotransmitters that you should be familiar with are like serotonin, dopamine, endorphins, and norepinephrine. If you still don't know these by memory, please go back and study these. Serotonin relates to the feel-good neurotransmitter. Dopamine also facilitates movement. Endorphins are associated with natural, like, painkillers, and norepinephrine just suits you back down to a more calm state. So please understand that these neurotransmitters are the neurotransmitters that secrete in the out the terminal button, and it is what is transcribed, or is what is acquired by the dendrites from the other neuron, and then it is sent to the soma to transcribe message. So moving on. Now we also need to talk about agonists and antagonists. We're almost done with this slideshow. So agonists and antagonists they differentiate vastly. These neurotransmitters are how we experience certain sensations, such as happiness, sadness, and anger. Many drugs can mimic euphoria, alertness, and other sensations by acting as agonists. So agonists mimic effects, they make things happen. Agonists are chemicals in drugs that can bind an active receptors in the synapses, thus providing the associated feeling of that certain high that people get into, like cocaine, heroin, and nicotine. So that is the high you feel. However, reuptake can be slowed, and these drugs can be overused and deplete the neural or the natural state of neurotransmitters, so leaving you feeling worse afterwards and the need of more drug usage. So this leads to addiction. So agonists slow down things, they stop things from happening. So antagonists halt, agonists go. So that's a very good way to differentiate. So agonists excite, antagonists do not. They actually halt. So what's worse, some drugs that act as antagonists, chemicals that bind and do not activate the receptors, sometimes permanently? I think that in both cases, agonists and antagonists are extremely bad. We should just use it to medical professions, so that way we can come up with certain medical uses for them, but not necessarily the way that they're used in the streets. So, viewing brainwave activity, right? We have talked at times that the way that people view brainwave activity is through lesions. Lesions is the destroying of brain cells. Believe it or not, at some point in time, people did get engaged in destroying certain regions of animal or rat brain parts, so that way they can know what certain parts are associated with what function. So it was very, very, very interesting, but nonetheless very, very revolutionary. So some other imaging methods or other methods that people have engaged on to be able to tell or study the brain more, we have talked about the EEGs, the electroencephalograms. We have talked about the CT scans, the computed tomography scans, which are more like x-rays. We have also talked about the PET scans. We've talked about the MRIs and the functional MRIs. If you still do not know the big difference between these, I suggest you go back and please review those. They're not too difficult to understand. Go watch some videos and it'll be easy to differentiate. But this leaves us with our last slide. So if you guys are having trouble just knowing the difference between these scans, the electroencephalogram is the picture that is right here. That is an electroencephalogram. It's used to measure brainwave activity. A CT scan is an X-ray that reveals brain damage, or it gives you a picture, a 2D picture. A positron emission tomography is it actually shows brain consumption of a chemical field that allows you to detect certain regions that are affected in the brain. It's something that physicians stay away from a lot, and that is typically left for last resorts. An MRI is an updated version of a CT scan. It just really gives you an updated and very detailed picture of how the brain looks. So it puts the head in a strong magnetic field that aligns spinning electrons of the brain molecules, and then it disorients them with a radio wave. And when they return to normal, it gives a detailed picture of the soft tissues. So a functional MRI takes an MRI scan and also tracks the blood flow to reveal activity in brain areas. So if you have any questions about this lesson, guys, just please ask me. I will be logged on this afternoon to answer any questions. But this was another review that we have needed to go through. If you have any difficulties, just give me a question. I am here to help, and have a great day.